Is it visible?
Yes, we can see it.
Is it visible, ma'am?
Yes, it is. You can start whenever.
Okay. A very good morning to all of you. Thanking you for giving the opportunity to discuss the emerging topic in International Conference on Business, Marketing and Technology in Canada, that is ICAPMOT 2025. I am Dr. Rupam Srivastava and going to discuss about the topic, the role of plasmonics in modern PCF-based optical sensors under supervision of Dr. Yogendra Kumar Prajapati, professor at MNNIT Allahabad, Prayagraj. Currently, Dr. Prajapati is unable to connect due to some reasons, so I'm going to discuss about the topic, the role of plasmonics in modern PCF-based optical sensors. So the motivation behind the topic is, as we know that sensors are required to gather accurate and timely information about various phenomena or substances in our environment, such as healthcare, environmental monitoring, food industry, etc. And what should be the characteristics of the sensors? Sensors should be highly sensitive, miniaturized, capable of early detection, portable, cost-effective, and easy to handle. So the traditional methods for in sensing applications, there are various traditional methods in sensing applications like liquid chromatography, flow cytometry, ELISA, that is enzyme linked immunosorbent assays, PCR, that is polymerase chain reaction, RT-PCR, etc. Although these methods are robust and widely used, but these methods also suffer from some major disadvantages, like these are expensive and these methods require tedious sample pre-treatment. Detection cost is also very high, and very experts, that is specialized persons, are required to use the equipment, and these methods are also time consuming. So whereas if you talk about the surface plasmon resonance based sensors, these sensors has emerged as a powerful tool for wide range of applications. And these methods are because these methods offer several advantages, such as these methods offer um, like label free detection. Label free detection, these are very sensitive, and these offer real time monitoring. So to overcome the limitations offered by traditional methods, the surface plasma resonance based sensors are the potential and emerging candidates. So they are due to various advantages. So SPR-based sensors are used in several fields. Like these are used in the field of medical diagnostic, food detection, glucose monitoring, environmental monitoring, telemedicine, chemical sensing, bioanalyte sensing, etc. So if you talk about the optical biosensors, now I'm going to talk about the optical biosensor. So optical biosensors are a sensor or a device that utilize optical principles and techniques for detection and quantification of biological or chemical analytes. And we can see in the figure 1.2, the key components of the optical biosensors are BRE, that is biorecognition element, optical transducer, and signal processing unit. So what is BRE or biorecognition element? This is the sensing element of the biosensor that is responsible for selectively capturing the analyte. And the BRE can be any enzyme, antibody, bacteria, cell, tissue, nucleic acid, etc. And optical transducer, so we know that transducer is a device which can convert a kind of signal to another. So here optical transducer means that it converts the interaction between biorecognition element and analyte into a measurable optical signal. So this is the role of optical transducer here. And after that, signal processing unit. So the once the signal is detected with the help of photodetector from optical transducer, then signal processing unit is used to for the analysis of that signal. These are the basic components of optical biosensor. And transduction method, several transduction methods can be used, like absorption, fluorescence. Here we are talking about the surface plasmon resonance based optical sensors. Now, what is surface plasmon resonance? So first of all, a term is coming like plasma. What is plasma? So if you talk, first of all, if you talk about the plasma plasmon are the basically electrons which are present at the uh, metal and dielectric surface. The plasmons oscillate at the boundary between metal and dielectric surface. So if it try to understand like this, that when light is propagating from denser medium to rarer medium, then and at an angle greater than critical angle, then TIR, that is TIR phenomena occurs. So when we place a thin film of metal between denser medium and rarer medium, then what happens? The complete reflection of light does not take place, but an evanescent field also generates. So here the what happens, that when the we are talking about the evanescent field, that is a part of light. So when the phase of that evanescent field and the electrons at the surface of metal, that is surface plasmons, becomes equal, then surface plasmon resonance condition occurs. And we can measure this SPR in two ways, that is angle integration method and wavelength integration method. In angle integration method, a monochromatic light source is used, and we vary the angle, the incident angle of the light. And in wavelength integration method, a polychromatic light beam is used and the wavelength is varied. So for surface plasmon resonance based sensor, several coupling methods are used. Like uh, one of the most basic or traditional method is prism based surface plasmon resonance. That is that utilizes Pressman configuration. So in this, what happens? A prism is used, and over which a plasmonic layer, that is plasmonic means any metal like gold, silver, thin film of any metal can be used. That is plasmonic layer. And what happens when the propagation constant of the surface plasmon wave, that is SPW, that is propagating along the metal dielectric interface, is equal to the propagation constant of the evanescent field of the light that is penetrating at the interface between the metal dielectric interface. Then when both becomes equal, then surface plasmon resonance condition occurs. And what happens at this stage, most of the energy is transferred to the surface plasmons, and so at that particular angle that is called as resonance angle, we got a notch or dip. So that is called as resonance angle. So this is the conditions are the condition for the surface plasmon resonance is given here. Although these mobile sensors are very robust, but they also suffer from lot of limitations, such as they are bulkier because they have very heavy mechanical and optical components. And so to overcome these limitations, we use fiber-based surface plasmon resonance sensor. So first of all, uh, actually, I am going to discuss about the photonic crystal fiber-based surface plasmon resonance sensor. So first of all, we should know that what is PCF or photonic crystal fiber. So PCF is also like a conventional fiber, but with some different characteristics. Like it also has a core and cladding, just like a conventional fiber, but its core and cladding consists of several air holes arranged in a periodic manner, and they can be either hexagonal, square, triangular. In various uh, arrangements can be possible. So and also, uh, what is the advantage of photonic crystal fiber over conventional optical fibers? The PCF offer design flexibility and possibility to control light propagation, and also it offers single mode propagation, and due to which high evanescent field with very high strength can be obtained. These are robust and cost effective, and these are also lightweight and portable as compared to conventional prism based optical sensors. So there are basically two types of photonic crystal fibers. One is index guiding PCF and other is photonic band gap PCF. So index guiding PCF comprises a solid core, and it follows the principle of total interaction reflection. Solid core. What is the core? The material, the background material generally is used as SiO2 silica, and the and the holes are made from the air. That is air holes. So index guiding PCF comprises a solid core, whereas photonic band gap PCF comprises a hollow core. That is core is made of hollow means there will be some air. So, and here the principle is different. That is, light is guided through the photonic band gap property in the photonic band gap PCF. And there are also different sensing methods can be used in PCF-based SPR sensors. that is internal sensing method and external sensing method. In internal sensing method, the plasmonic material is deposited inside the air hole, like we can see in figure 1.7a. Inside the yellow color represents gold or plasmonic material that is inside the PCF, that is inside an air hole. And in external sensing, we can see in 1.7b, the deposition of metal is at the outermost portion of the fiber. We can see in the orange color, uh, that is TZ, that is thickness of gold here. So what is the difference? So the major difference is, one thing is that deposition of metal, and also sample can be where the sample is going to be placed. In internal sensing method, the sample is being placed in the inside the air hole, and external sensing method, the sample is placed outside of the PCF. These are some performance parameters of the PCF-based SPR sensors, how to measure the performance. So basically, one of the fundamental parameter is confinement loss, which is given by the formula. And here, the imaginary part of effective refractive index is used in order to calculate the confinement loss of the fundamental mode. And other is wavelength sensitivity. And here, wavelength sensitivity is computed with the help of confinement loss. And amplitude sensitivity is also computed with the help of confinement loss. And resolution, other parameter is resolution. And methodology, finite element method is used to to analyze these parameters. We will talk about the software later. These are reported works from the year 2009, some of the reported work from the literature, which I mentioned here, from 2019 to 2024. Here, various kinds of PCF structures are used for different applications. Different types of arrangement of air holes are also used in different reported works for different applications. So before experimental work, we go for simulation, because simulation provide a robust design. So in order to simulate, console multiphysics software is used. It is a general purpose of simulation software for modeling and simulating physics-based problems. Uh, it is used across various engineering, scientific, and manufacturing fields. And what are the key features of COMSOL multiphysics? It is a multiphysics modeling. It is based on the finite element method, that is FEM. It offers wide range of application. It can be used in wide range of applications. It is very user-friendly interface, and it has simulation applications, and it also has interfacing products. So what is the modeling of workflow? First of all, we set up model environment in the console multiphysics. Then we build geometrical objects, which kind of like PCF based SPR sensor. So first of all, we will build the geometry of the PCF according to the given dimensions. Now we will create the definitions. Then we will assign the material properties, like air holes will be assigned air properties, and plasmonic metals will be assigned their properties. Then after specifying material properties, we will define physical boundary conditions. Then we will create the mesh of the whole geometry. Then after meshing, we run the study. And after run, we get the result, and then we can process the results according to our we can process different results, like confinement loss, wavelength sensitivity, amplitude sensitivity, resolution, FWHM, that is full width at half maxima, etc. These are the recent PCF-based SPR sensors using FEM performed by our research group. So in this, we can see here a D-shaped PCF sensor, that is external sensing-based approach. This PCF utilizes SiO2 as background material, and we can see here in figure 1.8 that in the zoomed version, the yellow color area tells about the plasmonic material, that is gold, and above this the bioanalyte is placed. And the parameters of different uh, geometry are given in this table, in the given table here. And to in order to calculate the if refractive index, wavelength dependent refractive index of silica and gold, we use the Selenium equation and Roux-Lorentz equation respectively, given by equation 1 and 2. So first of all, we will discuss about the geometry. First of all, a D-shaped PCF sensor will be used. Then a rectangular groove is utilized in order to deposit the metal and in order to place the analyte in this. Why? Because by incorporating this rectangular groove, the distance between core and metal will be uh, reduced, and the electric field strength will increase, due to which sensitivity will be increased. This is the concept behind this proposed SPR-based PCF sensor. So by analyzing the structure in FEM-based console multiphysics software, we get the results like core mode, coupling mode, and SPP mode. Figure A represents core mode, B represents coupling mode, and C represents SPP mode. SPP mode is the mode where the at the resonance condition, maximum of the light is transferred to the surface plasmons. So that is called the resonant wavelength. This is the wavelength integration approach. Okay, so maximum light will be transferred to the surface plasmons. So here we will get the maximum loss at the resonant wavelength, that is at approximately at 1.38 micrometer, 1.37 to 38 micrometer here in this figure, 4.3. 4.3 is representing the confinement loss characteristic curve and also the dispersion relation. So the resonance condition is also known as phase matching condition, and at this the real part of the SPP mode and fundamental mode becomes equal, and maximum loss is obtained at this point. Now, based on the confinement loss, uh, here the analysis performed for the analytes ranging from 1.30 to 1.40, and the loss curves for different analytes are depicted in figure 1.10, and corresponding amplitude sensitivity is also represented in 1.11, and these all results are tabulated in further slide, in the next slide. Here we can also measure different parameters like FOM, that is figure of merit, and full width at half maxima. And this proposed PCF structure also been used for the for the application of malaria disease. That there are different stages of malaria, that is wing stage, trophozoite stage, and Sjögren's stage, and that can be detected with the help of red blood cells. The concept behind this is that uh, when the malaria disease is occurring through different stages, the refractive index of the blood will be also be changed accordingly due to variation in the concentration of those bacteria. So uh, according to different stages, we got the loss characteristic curve for different stages as mentioned in 1.13 A, B, and C. And the here geometrical optimization also have been performed for gold layer variation and the two largest air holes as mentioned in this figure. If we go back, we can see here the diameter represented by D here, air hole. So this air hole and the thickness of gold layer is optimized in order to get the optimum result. So the result of that uh, optimization is presented in figure 1.14, A and B. Then these are the comparisons of reported work and our proposed work. So we can see here that we got the sensitivity of 67,000 nanometer per array, that is highest among all, and also very finer resolution. Resolution should be as low as possible, and figure of merit should also be high. And this is the comparison for malaria disease detection. That we can see here also that for ring phase, trophozoite phase, and Sjögren's phase, our proposed structural possesses the maximum wavelength sensitivity. Now the second proposed work is uh, here. In this, we use two-dimensional material, that is MAXine and MOS2. MOS2 in order to improve the performance parameters, because 2D materials also play a significant role in improving the performance parameter of PCF-based SPR sensors. And here we perform the analysis for tuberculosis detection, and the structure comprises of SiO2 as background material, and uh, over which MOS2 layer is deposited, and which is acting as field enhancement layer, and over which magazine layer is incorporated, which is acting as a BRE layer, that is biorecognition element. So these figure 1.16 A, B, and C represents corresponding core mode, coupling mode, and SPP mode, and the geometrical parameters are given in the table here, which are in the order of micrometer, and corresponding confinement laws. For we have considered here three cases. First case is the PCF-based SPR sensor with only plasmonic material, that is gold, that is represented in figure 1.17A. Then in B, we took plasmonic material along with MOS2. So he can see that in from figure 1A to B, the sensitivity has been increased. The shift has been increased from 120 nanometer to 130 nanometer. And in the third case, we took gold, that is plasmonic material, MOS2, and magazine. So that magazine is used here as the BRE, that is biorecognition element. Here the shift again increases to and reaches up to 220 nanometer. So according to these results, we got the maximum sensitivity for the third proposed structure, that is along with plasmonic material gold, field and enhancement material MS2, and BRE material exim, that is probe 3. Here we got the uh, maximum sensitivity of 45,000 nanometer per RIU. And different parameters like resolution and WHM are also tabulated in this table, and the corresponding um, report and the previous works which has already been reported in the literature also been compared here in this table. What are the future implications of plasmonic PCF sensors? So to improve the overall performance, we can utilize new emerging BRE materials, that is biorecognition materials. Also, to improve the detection limit, we can use different two-dimensional materials. And nowadays, the machine learning approach has been widely used. So, in order to effective utilization of the different structures, we can use the machine learning approach also. And combining the machine learning approach with our proposed work can open new possible study window for PCF sensors in near future. So this is the summary what I have covered. I have covered basics of optical sensors, that is components and applications, surface plasma resonance concepts and its application in sensing. I have also covered the conventional SPR sensors, photonic crystal fiber based SPR sensors, their methods of different sensing approaches like internal sensing and external sensing in PCF based SPR sensors, different kinds of reported PCF based SPR sensors in the literature, and I have also discussed about some of the works performed by our research group and its future implications. Now, my sincere acknowledgement to Professor Yogendra Kumar Prajapati, Department of Electronics and Communication Engineering, Motilal Nehru National Institute of Technology, Allahabad, Prayagraj, and Lab for Photonic Devices and Communication, where I have I have completed my PhD under supervision of Professor Yogendra Kumar Prajapati. Thank you, everyone. Now, if anyone have any query, then please you can ask.
Yes, thank you so much. I apologize, I didn't introduce you before you presented. So let me do that now, and then we can have the questions after.
Dr. Rupam Srivastava is an assistant professor at the Center for Internet of Things, Madhav Institute of Technology and Science, Dean University, Gwalior, Madhya Pradesh. Her research areas include surface plasmon resonance-based optical sensors, utilizing prisms, optical fibers, and photonic crystal fibers. Dr. Srivastava's work supports the advancement of smart technology and their practical application in various sectors. She is presenting this work on behalf of Professor Yogendra Prajapati, Motilal Nehru National Institute of Technology.
All right. So
Thank you. Thank you for. Okay.
Yeah, of course. I see someone has raised their hand. I can first ask you questions, and then if there are other questions in the chat, I will allow them to speak. So the first question is, how does the integration of SPR enhance the sensing capabilities of photonic crystal fibers?
Okay. Uh, Actually, SPR offers label-free detection. Like in general, uh, traditional methods, as I discussed in the earlier slide, some um, some labels are required in order to detect the analyte or sensing medium, while SPR offers label free detection. So it is the very good advantage of SPR. And PCF, if we integrate the PCF with SPR, then PCF offers, it is